In Lecture D, now we're going to focus on what the T cells are involved with. So remember earlier in the chapter we said that you have both the T lymphocytes and B lymphocytes working. T lymphocytes are more innate, or they are innate immunity, and they're more non-specific. So cell-mediated immunity. With this, we know that this is the first reaction of the immune system. It involves different classes of T cells. So we have the T helper cells and cytotoxic T cells. And then of course they come by other names. They respond again when cells have something inside of them, so intracellular pathogens. So we know that viruses have to hijack the host cell to take over, and we know that some bacteria will be intracellular. We know that cancer cells are diseased themselves, and anything from a transplanted organ that is non-self, and we've talked about that before, the antigens are non-self, the body considers them to be foreign. So all of those are a wide variety of different things that T cells would go to try to fight off with cell-mediated immunity. So we know again that T cells are formed by hematopoietic tissues in the bone marrow, but they leave the bone marrow, they go to the thymus gland, and there in the thymus gland they become what we call immunocompetent. So each one of them know exactly what specific antigen. They start off naive, they don't know what to fight off, but then they begin to recognize the specific antigen, they see it, the enemy face to face, and they'll begin to create clones of themselves. So the different clones become part of our immune system. There's only a few of those that exist in the body at any given time, so some of the T cell clones are capable of recognizing and responding to the pathogens, where others are still immature and not. So we know that with the thymus gland creating the hormones that are involved with T cell development, it also, with the reticular structure of the gland itself, screens the cells, mediates when cells need to be when they need to undergo destructions, and then the ones that can't recognize foreign antigens, of course, that's part of the self-test to make sure that they don't attack self cells, only foreign cells, those would be destroyed also. So number one, the first part of the test is to make sure that the T cells know what to attack, and then they create clones once they have that specific antigen that they will respond to. The second part of that is that they have to only attack non-self cells. So any cells that are self, you wouldn't want the T cells attacking. So once all of this process has taken place, the term is immunocompetent. So these are mature cells that can mount a response to foreign antigens. We know that we do have mature naive T cells that will migrate to the lymphatic organs, especially the lymph nodes, but we also said they reside in the spleen to await foreign pathogens, foreign antigens to be encountered. So with T cell maturation, we know that the hematopoietic tissues in the bone marrow, the cells undergo the lymphoid line, then they are in circulation, they're delivered to the thymus gland, they're in the thymus gland, they will test themselves, so they have to respond to particular antigens but not respond to self antigens. So the ones that are self-reacted will be destroyed. Some of the other ones that are mature T cells will eventually then wind up in the lymphatic organs like the lymph node, and there they'll actually wage one-on-one war with the pathogen. As far as the response of T cells, we know that certain antigens that are immunogens, these are the ones that actually cause or generate an immune system response. So we know again that our own cells have self-antigens, those flags that fly on our cells so that the immune cells can recognize what is self and what is not self. There are small antigens that are immunogenic because they're attached to protein characters, so they still are foreign. they're usually by themselves not recognized by the immune system, but when protein carriers pick them up, these haptens are the small antigens that will create an immune response. So some examples of haptens will be detergents, pet dander for some people, different grains and grasses in the air, pollen. All of those are small antigens, and they're called haptens. The receptors on T cells are found on the surface of every T cell, so that receptor is responsive to a specific antigen. Then once it does respond, it becomes activated, so that unique portion where the receptor can bind is called the antigenic determinant. So it determines which antigen will match the receptor and initiate a response. We know that there's only certain pieces of the antigen that are bound to glycoproteins. So remember, glycoproteins are that glucose protein, the carbohydrate protein complex, and we said from biology class, MHC proteins, major histocompatibility complex proteins. These are like ID tags or fingerprints. And again, I describe them as like flags that mark our cells. So those MHC molecules are on all nucleated cells except erythrocytes, because we know erythrocytes lose their nucleus and they lose their organelles. So this makes blood products easier to donate than other tissues like the pancreas or like the kidney tissues because of the MHC proteins on our self cells. So we know that those MHC proteins are like docking sites, and they become, once the antigen is taken care of on the MHC molecule, then it's displayed by the antigen presenting cells over to the T cell. So then they know what to fight off. So major histocompatibility complex proteins, we said, are like ID tags, and we said you could think of them like the flags that fly. So they mark our cells or self cells from pathogenic cells or invading non-self cells. Two types of MHC molecules. We know that class 1 are found on the nearly all nucleated cells. They use and present endogenous antigens. So these are the ones that are for inside of cells. They're either foreign antigens that present and live inside of our cells, or it could be a self-antigen that's encoded by our own DNA. If it is a pathogen lives in the cell, we've already said that you have, of course, viruses and intracellular bacteria that would reside. So our normal self-antigens, the foreign cancer antigens that mutated DNA would make, and all viral antigens would be endogenous. And again, that's why we call it cell-mediated immunity. It's inside of the host cell. So you have some that are very nonspecific, and they're called cytotoxic T cells. So cytotoxic, they degrade and become toxic to the cytoplasm, and they kill these foreign cells. So they can only interact with class 1 MHC molecules. So class 1 found in all nucleated cells, or nearly all, and cytotoxic T cells are what recognize the antigen presented in a class 1 MHC molecule. Class 2 molecules are found only on APC cells. So your class 2 present exogenous antigens. Exogenous antigens are always foreign, and the MHC class 2 complexes are the ones that would respond to this. And this is where your T helper cells will generally interact with the class 2 MHC molecules. So here you see the exogenous antigen fitting in the class 2 MHC molecule, and it's on the surface of an antigen presenting cell. So this would be where, like, a macrophage as an antigen presenting cell digests the antigen, displays it on its surface, and then the T helper cell recognizes that particular antigen and becomes activated. So T cell activation has the following steps. Step 1 from the figure shows that you display the antigen in the MHC complex. So I think of the MHC protein like the hot dog bun, and we call the antigen that's presented there, that's the specific part of the antigen that is pathogenic, is considered to be an epitope, or if you think about it like a hot dog in the bun. Then of course our T helper cells, or cytotoxic T cells, will bind to that antigen. They undergo what we call co-stimulation, so they'll stimulate each other, both the T helper and the cytotoxic T cells. As they bind to the co-stimulator, they both are now activated. Then once they're activated, they'll proliferate into clones. Some of them become the effectors, meaning they carry out the war, they wage war on the pathogen. Another group of them become the memory T cells. So these are the ones that give you immunity for later use. So memory T cells respond much more quickly and efficiently once it's already been exposed to the antigen. So you don't need a co-stimulator once the memory T cells are in circulation. So your cytotoxic T cells, your T helper cells, each one of those play different roles. They do interact together, but each one is functioning independently of each other. So notice your T helper cells are non-phagocytic. They don't have cytotoxic abilities. They exert their effects by secreting cytokines. So these are chemical messengers that will activate and enhance other parts to the immune response. So in this way, that's why they're called helper cells, is because they, quote, help the immune response out. They're required for normal function of all components within the immune system. So again, some of the cytokines that we'll talk about are the interleukins that are much more part of the chemical pathway, the biochemical pathway. Our cytotoxic T cells, again, cytotoxic because they can actually kill other cells. So they remind us of the natural killer cells because they respond to any foreign antigen that's bound to a class 1 MHC protein. So they can detect any abnormalities within a cell that has a nucleus. So this, of course, our cytotoxic T cells are used to detect cancer cells, foreign cells that are introduced into the body, and then cells that are infected with intracellular pathogens, of course, like the intracellular bacteria and viruses. So here is where the T cell binds to the target cell at that particular receptor. Notice the MHC class 1 is involved with the cytotoxic T cell. Then in step 2, you notice it releases the porphyrin, which creates the pores in the target cell. Then it'll release the granzymes, and these enzymes from the cytotoxic T cell go to the nucleus to fragment out the DNA. Well, once the DNA fragments, of course, the cell is going to lose control and it begins to degrade and tear down. So then the cytotoxic T cell detaches from that cell and searches for another target cell, just like the one it previously destroyed. So cytotoxic T cells, you see in the figure, binding to a cancer cell. That whole process is going on. So again, it's releasing the porphyrins make the whole. Then it releases granzymes, which are different enzymes that are cytotoxic. They go to the nucleus and they degrade the DNA, and then the cell begins to break down and degrade. So the porphyrins again create the hole or the pore, and the enzymes can invade the target cell, go toward the nucleus, and then target the DNA as it degrades the nucleus of the invading cell, or the cell that's diseased. So in this section we talked about the cells that are involved in cell-mediated immunity. Cell-mediated is your T cells. Your T lymphocytes are involved with cell-mediated immunity. Describe when they respond. Well, remember, they have to know what to attack, so they have to be presented with the antigen. Remember, they mature in the thymus gland, and then they have to not respond to self cells. So in their maturation, we say that they're mature once they pass both parts to the test, and then they become immunocompetent. So we say they're immunocompetent once they know what to destroy, and they do not respond to self cells, and they've passed both parts to that test, and they now begin to clone themselves. Some of them become memory T cells, and some become cytotoxic T cells, and some are the helper T cells. The differences between antigens or the immunogens, self-antigens, haptens, and antigenic determinants. So we've already said antigens are anything that are non-self that can cause an immune reaction. Our self-antigens are our MHC proteins that mark our cells like ID tags. And then we said the small, not as large of an antigen, but yet there's small pathogenic substances like pet dander, detergents, cosmetic products. All of those are examples of haptens that would create an immune response in some people. If we describe the MHC complex, we know you have MHC1 and MHC2, and we differentiated between each type, and we looked at the functions of it, and we looked at what T cells respond to each one of those. Then we looked at endogenous and exogenous antigens. So endogenous, of course, these are cells that are infected or cells that are diseased inside. Exogenous antigens are the ones that are displayed outside of the cell. Both the processes for T lymphocyte activation, how they're cloned, their differentiation and their proliferation, those are described in the figures from your textbook, so that you have both the picture to look at and the information in the synopsis. The roles of both the helper T cells and cytotoxic T cells, we covered both of those when we looked at MHC proteins and which one responds to which. And then porphyrins, we said, creates the hole or the pore in the membrane so that the enzymes, a lot of times in some textbooks they're called granzymes, can invade the cell. And then where do they go? They go to the nucleus to destroy and fragment the DNA to destroy that cell. So that's why they can attack cancer cells, cells that have viruses in them, and cells that have intracellular bacteria. So this is the main thing with HIV. The HIV virus attacks our T cells. So I've said it many times that no one's ever died from AIDS, the syndrome, or just having the T cells be attacked by the HIV, but it's because they die from diseases that the T cells would normally fight off, because the HIV attacks T cells. Well, those are the ones that would detect an intracellular virus. So it renders it, literally this portion of our immune system, it renders it useless.